Okay, now this is the beginning of the rapid ID method, and this is actually a really fun lab. It uses a tool that actually is used in clinical laboratories to determine what type of intestinal bacteria a person might have. So if a person comes to the hospital and they present with diarrhea, vomiting, there's many different enterics that could possibly be the problem, and some are gram-positive, some are gram-negative. So to determine what type of antibiotic to give, you would run a test like this to narrow it down to, you could actually narrow it down to the species of the bacteria based on the results. Now, an enteric, like I said, is an intestinal bacteria. Now, a lot of bacteria are very common in the intestine, but some of them can cause disease. They can be opportunistic pathogens. And a list of the ones that we're possibly using in this lab are on page 107. E. coli is one, Citrobacter, Enterobacter rogenes. You can just see some of them by the name Bacter, refers to the enteric. Serratia marcescens, that red one is a possibility. Proteus vulgaris, Proteus mirabilis. Oh, so some of these we've already used in the lab, and some of them we have not. So when you do yours, you could have any one on the list in your group. So basically what you're going to be given is each group is going to be given an unknown. Now we're either going to have the 8 groups like we normally do, or we're going to have 7 groups. I forget how many actual kits they're going to give us. So it just depends on what the tech gives us in terms of what we have. Now, so what you're going to do is if you look on the left side of the page 107, there's what's called the IMVC test. I stands for indole, M for methyl, V for the VP test, and then C for the citrate test. Now, all of these you have done before. So what I'd like for you to do before you come to lab is go and research what these tests are. Like I said, the methyl red, the VP, and the Simmons citrate you did back in the fermentation of carbs. So go back to that lab, read up on it, look what you're testing for, write what the results should be, the different reagents you're going to use, so that you're prepared for it. Now, we haven't done the indole, and the indole is one that's actually not in your lab manual. But if you refer to your atlas, you'll be able to figure out what the indole test is going to be. So I want you to research those, write them down before you come to class on Wednesday. Okay, so what you're going to do is you're going to inoculate those 4 tubes. So we have the MRVP broth, remember, one for MR, one for VP. If you recall, the Simmons citrate had that green auger, which is a slant. And then the indole is going to be a light color, just like the nutrient broth. And so for those, you're going to use your inoculating loop, and you're going to inoculate those particular tubes. So remember, for the slant, you want to just run it on the top of the slant and not gouge the auger. But for the liquid, you can actually put it down into the liquid. Remember your sterile technique, you're going to be using your loop or your Bunsen burner, and which I'm not going to do, but you all know how to do your sterile technique. Now, I would like for you to look on page 108 first. Okay, on 108 it says that you're actually going to streak the plate. Well, you're not going to do that because that's going to be done for you already, because since we only meet once a week, you guys won't be streaking the plates. So you'll actually be provided with a plate. It'll have an unknown number on it. So you go ahead and just write that number down. That'll be the unknown for your group. Now, then it says determine the oxidase reaction. So that's something you're going to do before you start inoculating all the other tubes. For the oxidase reaction, all you're going to do is take your sterile loop, and you're going to get a loop full of the bacteria. Okay, just pick one isolated colony. And then an isolated colony means it's one colony out by itself. So don't pick where it looks like the colonies are all smeared together. Okay, and then for doing the oxidase test, we actually we have 2 different methods of doing it. One is this liquid right here, and you could actually put a drop of it on the plate. If we have the liquid, then you're actually going to put the drop on the plate after you inoculate everything else, because you don't want to contaminate the plate before you put your specimen to the other tubes. The other option that we can do is an actual strip. So it's a little paper strip, and you would actually pick up the colony and rub it on the strip. What you're looking for is an immediate reaction, and if it is oxidase positive, it will immediately turn blue. Okay, but don't be disappointed if you don't get that immediate blue color right away, because you actually want them to be oxidase negative, because this tube right here tests for bacteria that are oxidase negative. If they come up positive, then it might not be the correct bacteria, okay? And like I said, it is an immediate result. You don't wait 5-10 minutes and then it turns blue, because all of them will eventually turn blue over time, okay? So either way, if you do the strip, you do it first before you inoculate anything else. If we have the actual liquid, then you'll inoculate everything else and then put the drop of liquid on there to make sure you don't contaminate it. Okay, so after you've inoculated these tubes, then you're going to do the Entro tube. And if you look on page 108, is the procedure for the Entro tube. Now we will not do the API 20E, so on page 110 and starting on page 111, we're not going to do that one. Those are two of the same tests, so we're only doing one. Okay, for the Entro tube, it looks like this, and it says to pick a colony using the wire end, the pointed end of the wire. So what you have to do is open up both ends. There's the white end, and that's where the wire is, okay? And it's kind of a point. And so when you go to pick up the bacteria, you don't want to gouge the auger. So just scrape the colony off, okay, without gouging the auger, all right? And then it says hold the bent end of the wire, so the other end that came off the blue is this one here. And what you're going to do is you're going to pull it through, and see how I'm kind of twisting it as I pull it through, because I want to make sure that I get inoculum on all sides, okay? And then it says to put it back through. So you pull it through all the way, and then you send it all the way back. Okay, once again, we're just trying to get as much inoculum on everything as possible. Okay, then after you do that, there's a little notch right here. So here's the wire, and there's a little notch right here. You're going to put it to where it goes to that notch on the end of the tube itself, and then you're going to point it away from you, and you're going to break it. Okay, and it breaks right off. Okay, with that part that you broke off, you're going to poke holes in these compartments right here that have the little notch in them. Okay, so you're going to poke a hole in the green one, each one after that. Okay, you don't poke it in the paper on the bottom, you poke it in the side. Okay, and then you'll notice the last 4 compartments don't have those notches. Okay, and then we'd have a beaker of bleach to put that in. Then you're going to put your lids on both sides. Okay, and then you're going to put it in the incubator. Go ahead and label it with your group name, and then you put it in the incubator. Let's see. You probably set it like that in the incubator. Okay, so you have the little notches that you've poked the holes in. Label it with your number and your group name, and set it in the incubator. Okay, you don't want to stack them on top of each other. So you'll have the 4 tubes you inoculated plus this, and then you would have already done that oxidase test. Okay, so that's the procedure for the rapid ID methods.